Hi, good afternoon, and thanks both Jack and Audie for the kind invitation, and to all of you for joining RheumNow Live, a truly wonderful gathering of both kind souls and brilliant minds. I'm sorry I cannot be there in person, but I'm Jose Scher from New York, and I will be discussing with all of you some of the work in the field around microbiomics, pharmacomicrobiomics, as it relates to spondyloarthritis and rheumatic diseases in general. Here are my disclosures. And so I will be talking today about the significant progress made in the understanding of the microbiome and how can it affect the pathogenesis of inflammatory conditions, with emphasis on spondyloarthritis and psoriatic diseases, the interface between the gut microbiome and efficacy and toxicity of some of the immunotherapies, and then we'll very briefly summarize where the excitement is actually coming from, and also the innovative aspects of DMARD dependency on the gut microbiome, finishing with some of our own work on the pharmacomicrobiomics of methotrexate in rheumatoid arthritis. So, as an example of a prominent form of spondyloarthritis, and this can be obviously applied to AS and others. We've known for many decades that psoriasis precedes PSA in most patients, that about a quarter of people with psoriasis will develop a form of synovial enthesial disease, and that given the right genetic substrate, certain environmental exposures, obesity, mental biomechanical stress, and or infections can actually lead to an aberrant activation of the immune system during what we now consider the preclinical phase of PSA, and which in turn can advance to a more clinically evident arthritis form if perhaps a second hit ensues. And this is the overall topic of my talk today, meaning, do we sufficiently understand the roles of microbes in SPA and PSA pathogenesis, a potential determinant of the passage from preclinical into clinical arthritis, as well as the mechanisms that can perpetuate disease once established, including the interdependence with drugs that we prescribe in the clinic. Of course, you know, the ultimate question will be can we actually prevent inflammatory arthritis in this context? And B, is it possible to reset inflammation to a homeostatic state once disease is clinically apparent? So amongst those potential determinants of disease progression, there's the microbiome. And we've known for over a century or so that microbes and infections can trigger psoriatic and spondylo-like disease, classic examples being strep infections causing gutted psoriasis, particularly in children, and obviously reactive arthritis. For spondyloarthritis, there are now very many animal models showing that specific microbiota can trigger arthritis, sacroiliitis, IBD features, and psoriasis-like rash, whereas mice that are raised under germ-free conditions, that is, in a bubble without any microbes, actually do not develop the phenotype, right? And here's one such example on the SKG model, which is both IL-17 dependent, as you can see on the upper right panel, and also requires the presence not only of gut bacteria, but very specific types of bacteria to ultimately acquire the articular and cutaneous inflammation. This has been studied for almost a decade now. In humans too, multiple groups have shown that both gut and skin dysbiosis, this process that perturbs the homeostatic community composition of bacteria and other microbes in both the gut and the skin and other surfaces, can be found. This dysbiotic process can be found in nearly every single autoimmune and rheumatic process, whether we're talking about rheumatoid arthritis, psoriatic disease, psoriasis, lupus, spondylo, AS, the findings have been similar study after study. Inflammatory autoimmune disease equals dysbiosis in these surfaces and in the gut in particular. And of course, the biggest challenge to the field has been what to do about it and whether this has any mechanistic implications for disease. And so something similar has been recently observed in both non-radiographic axial SpA and AS. This is the Swedish group published a couple months ago. This was pointed out to me by Jack, where a gut dysbiotic process is significantly more frequent in both populations of non-radiographic axial SpA and in AS compared to healthy individuals. You can see on the left panel that that most dysbiotic processes were found in these patients. And importantly, on the right panel, the group further observed that the higher the degree of perturbation in this gut bacterial community composition actually correlated with worse disease activity, whether measured by BASTI or ASDAS. The higher the dysbiosis score, the higher the disease activity. Our group has published now 7 or 8 years ago with Carles, Ubeda, and others that patients with psoriasis and psoriatic arthritis have also a decreased gut microbiota diversity, and that decrease in diversity is characterized by a significant decrease in certain beneficial commensals such as Clostridia, Rhuminococcus, and others, as well as a decrease in protective metabolites, including short and medium chain fatty acids. And, why is this important, right? The answer is because we now know that it is the bacterial metabolites, such as the short chain fatty acids, and not necessarily the microbes per se, that have the immune modulating properties. We have witnessed this in work published several years ago, revealing that in the presence of specific taxa, otherwise indigestible dietary fibers, right, starch, are metabolized into fatty acids, including acetate and butyrate, which in turn can now activate regulatory T cells in the lamina propria via certain receptors. Doesn't really matter, but in this case, GPCR 46, 43, and others, and ultimately suppress downstream inflammation, such as in models of IBD and others, via decrease in Th7 T cell. And it's also been proven to be the case in hampering systemic inflammatory responses in murine models of inflammatory arthritis. It is true for CIA, collagen-induced arthritis mice, where acetate, you can see this in the graph, and propionate both ameliorate synovitis, and also for the KBXN serum transfer model, where both short-chain fatty acids and a high fiber diet can decrease inflammatory arthritis. And so the question is, can we modulate intestinal microbes or their metabolites in order to treat immune-mediated disease, and specifically spondyloarthritis and psoriatic disease? And of course, there are several ways by which this can be achieved, spanning all the way from fecal transplantation to the modification of the entire ecosystem and single strain, or even bioactive molecular approaches that are obviously more specific in nature. Now you target, certain molecules that are produced by unique strains that now are known to mediate the inflammatory effects on the host. So any one of these approaches are being studied and tried. For example, this is from Georg Schett's group, published recently as well, a proof of principle study in rheumatoid arthritis, where patients underwent an intensive high fiber diet regimen for about a month, which led to an increase in systemic levels of short-chain fatty acids. You can see that on the upper panel, which in turn also modified the gut dysbiotic process and concomitantly decreased in the lower panels levels of circulating pro-arthritogenic cytokines, including IL-18 and interleukin 33 as well. Certainly this merits further investigations to help us understand if the clinical phenotype can also be improved, and also the idea that this can be extrapolated into spondyloarthritis cohorts as well. And then, of course, the favorite of many, there is the fecal microbial transplantation, which is essentially the administration of a stool solution from a donor into the intestinal tract of a recipient. This can be achieved through many different routes and procedures. These include upper and lower endoscopy, enema, and most recently, even frozen cups. This procedure, as you probably know, has been FDA approved for the treatment of refractory C. diff colitis and has proven quite effective in related conditions such as IBD. Right? This is based on original data derived from successful amelioration of ulcerative colitis, in this case, in the FOCUS study, which was published in The Lancet about 5 or so years ago, and showing that a multi-donor intensive, which is daily, FMT approach was superior to placebo in achieving both clinical and endoscopic remission, which is no different from the biologic outcomes that we see using ustekinumab or TNF blockers, for example, just to give you a sense of what kind of responses are being achieved with FMTs. So the anecdotal evidence from a year or two ago revealed that it is possible to achieve drug-free remission in PSA, in this case, following fecal microbial transplantation for another indication, right? It was in a patient with C. diff colitis, which provided this proof of principle that modulating the gut microbial communities can indeed affect downstream inflammation in both skin and joint domains. And based on all of these animal and human data, we now have the results of this pilot and yet very important study named FLORA, which was a double-blind randomized placebo-controlled trial comparing the efficacy and safety of one FMT procedure, that's important, via upper endoscopy versus sham solution in peripheral PSA patients. So the primary endpoint was a little bit unusual. It was a composite outcome based on shared decision-making and determining treatment failure, both by the physician and the patient at week 26. So that would be one of the caveats. But interestingly, and although underpowered, there were only 15 or 16 participants in each arm, the results showed that the group that received the sham solution performed unexpectedly very well and significantly better than the group receiving FMT. So this is curious and relevant for a couple of reasons. In my view, the placebo group achieved ACR outcomes that were similar to those seen in biologic therapies, which is incredibly curious, right? You can see that in the right panel on the ACR 20s, 50s, and 70s on the gray bars. But second, the FMT group did rather poorly, which in essence supports the notion that modulating the gut communities does have an impact in systemic joint inflammation, at least in psoriatic arthritis. Whether this is related to donor, recipient, or specific microbes, we cannot say based on this study because there was no microbiome-based readout. But certainly more to come and much more to learn in the future as to whether or not this is the way to go. Switching gears, a few words on this tongue twister, pharmacomicrobiomics, which is this new discipline in many ways that investigates the effects of variations within the human microbiomes on the drugs that we prescribe. And this work's pioneer, again, in both the oncology field, but also in the IBD field. For example, you can see a couple of studies here that I'm showing that when you use machine learning algorithms, predicting which patients will achieve steroid-free remission in both ulcerative colitis and in Crohn's disease, you can see that this algorithm that incorporated gut microbiome features improved the predictive value and went from an AUC of around 0.6 using the clinical features only in each one of these cases to well above 0.83 when you incorporated microbial features to the machine learning algorithm. Similarly, our group, in collaboration with the Italian group led by Francesco Ciccia, looked at whether specifically IL-17 blockade could actually lead to a gut dysbiotic response by way of microbiome perturbation, that is fungal communities, with downstream aberrant activation of the immune system or response that could potentially help explain, and at least partially, why a small proportion, and emphasis on small, of patients develop IBD-like syndromes when exposed to IL-17 blockers. So we characterize patients with spondyloarthritis in this case, before and after either TNF or IL-17 blockade, and perform gut micro and microbiome analysis as well as shotgun sequencing. And curiously, not only IL-17 inhibition led to more significant changes in the gut bacterial composition. You can see that on 16S with the Clostridialis leading the charge, but also we found that specifically Candida expanded in about a third of patients compared to those that received TNF inhibitors, which is not surprising, right? It isn't surprising since oral candidiasis is found in some patients treated with IL-17 blockade. But these findings can also, in our view, contribute to our understanding of why a small group of patients that are treated with 17 blockers ultimately develop IBD, and essentially the question of, can we use the microbiome features to predict who amongst those will end up developing the adverse event. So finally, the question of, can any of these be applied to clinical decision-making, and the lessons that we are learning from methotrexate in rheumatoid arthritis in this case. So we've learned a significant amount about the metabolic fate of methotrexate, what happens once patients that are being prescribed with this drug ingest methotrexate. We know a lot because there's an established high, very high, individual variability of methotrexate oral bioavailability, which ranges anywhere from 20 to 80%, depending on the studies you look at and the populations of study. And its the absorption of methotrexate seems to be at least partially limited by gut epithelial transporters, which appear to be saturable at a dose of around 15 milligrams or so. But this is probably an incomplete picture, mainly because the field has established about 50 years ago, half a century and now, that methotrexate is effectively metabolized by intestinal bacteria, at least in mice, right? So this is very elegantly demonstrated by NIH investigators, Lorino and colleagues, who demonstrated that wild-type animals, animals that are raised under normal circumstances, can actually metabolize the majority of methotrexate that they get gavaged with into DAMPA, which is this known inactive metabolite of methotrexate. However, both in antibiotic-treated mice, you deplete the gut microbes, and under germ-free conditions, which is the same outcome, methotrexate remains mostly intact, right? You gavage animals with methotrexate, and most of it remains in the original form, and the inactive metabolite, it's not seen. And so this suggests, for the most part, that methotrexate does have a relationship with the microbiota in mice that can now modulate its downstream effect. So about a decade ago, we first found that patients with new onset drug-naive rheumatoid patients had a markedly distinct gut microbiome dominated by a specific microbe. Doesn't really matter, Prevotella copri. But then asked the question as to whether or not methotrexate actually led to perturbations in the overall microbial composition, and found that although the number of species, or OTUs, diminished very dramatically in those receiving vancomycin, and you can see that in the red bars, those rheumatoid patients on standard care methotrexate did not have a significant change in their gut bacterial OTUs or species here in blue, suggesting that methotrexate does not directly affect the amount and relative abundance of various bacterial tags. I mean, this off-target effect or antibiotic effect was not seen at that time. So fast forwarding to this past year, we just published on how the baseline gut microbiome and its functional genes, very important, are different rheumatoid patients that respond to methotrexate compared to the microbiome of those that do not. Moreover, we've established for the first time that human gut bacteria can indeed metabolize methotrexate, and by using random forest modeling, we're able to predict with a high degree of confidence future clinical response, right? So previously proposed clinical pharmacogenetic models were of relevance, but this model appears to surpass the previously proposed models, suggesting that the gut microbiome can potentially be used for precision medicine approaches in inflammatory arthritis in general. So here's the study design. We'll go through this quite quickly, but we first enroll a discovery cohort of patients with new onset rheumatoid patients, arthritis that is, and follow them longitudinally to classify them as either responders or non-responders based on a very stringent definition, dependent on DAS28 reduction. And to do so, we collected baseline microbiome, subjected these samples to both 16S and shotgun sequencing, with the goal of understanding if any differences could be characteristic of non-response. And in fact, we did see initially that patients that did not respond to methotrexate had a higher gut microbiota diversity, and also a differential abundance of several taxa when compared to samples from responders. But more importantly, we think, is that we found differential bacterial pathways and gene orthologs in the treatment microbiome, in the pre-treatment microbiome of the training cohort of these NORA patients, new onset rheumatoid patients. Briefly, we observed significant differences in multiple gene orthologs within the gut genome of responders versus non-responders to methotrexate. We then applied this random forest to a training cohort and found that about 38 out of more than 450 gene orthologs were highly predictive of response. And finally, validating the model in an independent cohort and found that the metagenomics model, similarly to what we see in the IBD literature, was significantly more robust in predicting methotrexate response than a previously formulated clinical pharmacogenetic model. So taking together, here is the imperfect but somehow logical model where we are pursuing to better connect all these moving dots. In the proportion of rheumatoid patients, we believe there's a relatively low abundance of microbes that can encode for methotrexate metabolizing enzymes, so that the majority of ingested drug is absorbed in the systemic compartment and then act downstream to mitigate joint inflammation. But in other patients, they may actually carry a higher abundance of these enzymes, biotransforming methotrexate within the intestinal lumen, and ultimately preventing its absorption and clinical utility. But as I said, the model is quite imperfect because since in collaboration with Renuka and Pete Turnbow at UCSF, we just showed that methotrexate at higher doses also have off-target effects on human gut microbiota in cell cultures, in mice, and also in patients. This was less so on the overall community structure of gut bacteria, but rather impacting the expression of conserved metabolic pathways and their activity in gut bacteria. And importantly, some of these shifts also associated with methotrexate response, suggesting that the off-target effects, bacteriostatic, if you will, of methotrexate on the gut microbiota can also have consequences for immune function and response. So to summarize, I hope I was able to articulate the fact that there's emerging data implicating gut human microbiome as determinant of both pathogenesis, spondyloarthritis, and drug efficacy and toxicity in these diseases and related conditions. That multi-omic platforms allow for the generation of predictive tools and also for mechanistic studies, and finally, this idea that we can now exploit the microbiome and consider it for an effective and rational way of initiating anti-rheumatic disease drugs, and the potential to be incorporated into precision medicine strategies. And with that, I'll acknowledge every single one of the people that have been doing the really good work in our group and our collaborators elsewhere. And then a couple of post-test Q&A. I hope you learned something here. The first one would be the following: gut microbial perturbations or dysbiotic processes have been associated with which of the following rheumatic diseases? Give you a couple of seconds to respond.
There you go. All the above. All right. I think that's what we're.
And I hope I was able to show data that suggests or actually confirms that every single one of these rheumatic diseases have a dysbiotic process, at least at the onset of disease. Second one is gut microbial communities and metabolites are being used as therapeutic approaches in autoimmune and rheumatic conditions. Is this true or is it false? I'll give you 2 seconds to respond. And based on the FLORA study and others, I hope your answer was number 1. And with that, I'll thank you so much for your attention. Happy to take questions during the Q&A, and I appreciate your attention and the invitation once again. Thanks.
We're going to hold off on questions for Dr. Scher. He's going to come back at the end of the session, and we have a bunch of questions for him at that point. So if Eugene, you could hold on, that would be good. All right, our next speaker is Dr. Philip Meese. Philip comes from Seattle, where he is well known locally, regionally, internationally. Philip sort of started this whole problem of clinical trials and spondyloarthritis many years ago. He hasn't stopped. He continues to lead the way. Philip's going to talk to us about a new hot topic, axial psoriatic arthritis. Philip, thank you for coming.